Hi everyone. Today we are very happy to host this online seminar. We are very happy to providing an opportunity to share the new knowledge with the friends from the different countries. And tonight we will cover two main topics. The first one is presented by Dr. DJ Hsieh, and his topic is learning from nature, the best approach from regenerative medicine. And he will introduce for us what is regenerative medicine and how to achieve it, and the novel technology and the future approach. So the second topic is a serial tissue management technique for restoring a alveolar tissue defect and environment around the dental implants. Is by our Dr. Lee, Director of Association of Asia Reconstructive Dentistry, and and he will to share for us how to use the novel biomaterials in the in the in the dentistry in many clinical cases, and to achieve a real tissue engineering. So the first we are welcome our Dr. DJ Hsieh. Let's start it.
Okay. Good evening, everyone. Let me share my windows. Can everybody see it?
Okay.
Okay. Right. Okay. Good evening. It's a great honor to be here, discuss with you guys the regenerative medicine. My name is DJ. I'm the CEO of ACRO Biomedical company, and this is my background. I received my PhD from Cell and Molecular Biology at State University of New York at Buffalo, and I was an associate professor in Kaohsiung Medical University. And from the background, I saw my student, Dr. Li Mengjie, also join us. He is one of my students, my favorite students. Okay, the company was founded in June 2014. Our company is located in Kaohsiung Science Park, and we are dedicated in the research and development of nature collagen scaffold biomaterial for tissue engineering and regenerative medicine. And so far, we have received 69 international patents, and there are more than 20 pending patents. And so far, we have received numerous FDA approval from US, Taiwan, Singapore, Philippines, Vietnam, Thailand, and India. And we are a public company. We were listed in Taipei Emerging Stock Exchange Market since July 2021. And our company is focused on tissue engineering, cell therapy, and organ engineering. And tonight I will talk about our supercritical CO2 extraction technology and the use of this technology in regenerative medicine. And Dr. Li Mingke will discuss with you how the material can be used for dental application. And as you may know, that the tissue engineering regenerative medicine is a fast-growing industry. The annual compound growth rate is 26.1%. And the reason for this fast growing is because there is a severe shortage of donated tissues and organs around the world. According to WHO statistics, less than 10% of the demand in organ transplantation are met. That means more than 90% of the patients are still waiting in line for donated tissues and organs. So our mission is to apply biomaterial from animal source to regenerate human defected tissues and organs, and ultimately become the global leader in tissue engineering and regenerative medicine. And the vision is that one day people will not have to wait in line for donated tissues and organ, and tissue and organ transplantation can become easily available and affordable. So this is our platform technology. What we do is to put the porcine tissues and organ into this supercritical CO2 extraction system to remove all the immunogen, which include the cells, fats, and non-collagenous protein from animal tissues and organ, and leave the natural collagen scaffold material intact as biomaterial for human tissue and organ regenerations. And we like to call this technology the Holy Grail technology for the preparation of biomaterial for tissue engineering and regenerative medicine. The reason is that it's a minimum manipulation process and it does not disrupt the natural collagen scaffold. As you can see here is a piece of porcine skin. When the supercritical CO2 process the skin, all the cells are completely gone. It's removed. And if you check under electron microscopy, you can see that the natural collagen scaffold remain intact. There's no signs of damage at all. And in the process, there's no chemical solvent involved. And it can kill bacteria and virus at the same time. It's an environmental friendly process. You can recycle the CO2 and reuse it. And it can be applied to various tissues and organ, and also tissues and organ from various animals. You can use the porcine, bovine, equine, ovine, or even human tissues and organs. And it can reduce the immune rejection quite dramatically. And here are some of the SEM photos of the ECM structure after supercritical CO2 process. As you can see here, even though they are all collagen scaffold, but the collagen alignment and the mechanical strength varies from tissue to tissues, from organs to organ. So here there is an important take-home message. That is, when you have a defect on the skin, you will not use the bone to fix the skin defect. Also, if you have a defect on the cornea, you will not use the skin structure try to fix the cornea defect. That means you have to use the exact material to fix the exact counterpart of the defected tissues and organs. So our goal is to use the porcine tissues and organ to fix human defect from head to toes, from outer skin to inner organs. And eventually we can regenerate the whole organs so that people can replace any tissues and organ by this technology. And ultimately, people can live up to 120, 150. And here are some of the milestones after our company established in June 2014. We performed the world's first canine cornea transplantation in 2016. And the pet dog went from blind to visible within 30 days. So Discovery Channel actually came to Taiwan to feature the story and broadcast it in North America. And in 2017, we out-licensed the cornea product to Oculus Biomed in Australia. And we also signed an exclusive distributorship with Marubeni in Japan. And in the same time, some of our other products received US FDA, Taiwan FDA, and Singapore, Vietnam, Philippines approval. And in 2018, the cornea product was initiate the IRB human clinical study in Taiwan. And in 2019, we have a collaboration with NASA to join the astronaut osteopenia. And we also went to Hangzhou, China, to participate in the innovation competitions, and we got second prize. And in 2020, we received more FDA approval and patents, and we write some manuscript and submit it to international journal for publications. Due to the pandemic, we cannot go anywhere, so we publish papers. And in 2021, our company was listed in Taipei emerging stock market. And in 2022, more approval in the products. And 2023, we signed exclusive distributorship with DKSH for the collagen matrix and bone graft in Vietnam market, and also received more FDA approval and some awards, especially these Pharmaceutical Technology and Research Development Award in 2023. And here are some of our patent portfolio. Our patent cover the corneal transplantation, cartilage regeneration, aesthetic microsurgery, skin slicing and corneal slicing device, and also organ regeneration, bone regeneration, and hair regenerations. And the red dots represent granted patents, and black dot represent pending patents. And we have patent cover Taiwan, US, China, Japan, Korea, India, Hong Kong, Singapore, and European communities. And let's look at the product from the porcine skin. We have this collagen raw material, which has a registered device master file in the US FDA, and a series of scar care products, and also skincare products called ACRO. And we have product for the eye care called eye mist. And the collagen matrix is a decellularized porcine skin for wound healing. And the collagen membrane is from the porcine dermis, decellularized for dental applications. And because it's a dermis structure, we cryo-grind it into particles. And it's an ADM scaffold material for wound healing and also for hair regenerations. And you put those particles into the prefilled syringe together with collagen slurry, it becomes ADM paste for wound healing and also for wrinkle injections to make people 20 years younger within 10 minutes. And a cellular dermal patch is for internal organ operations. And the collagen matrix material intended for burn wound, trauma wound, diabetic ulcer wound, and surgical wounds. And they have US, Taiwan, Singapore, Vietnam, Philippines, Thailand, India approval. And it comes with many different shape and specifications. And collagen membrane for dental guided tissue regeneration and guided bone regeneration, and got Taiwan, Philippines, and Thailand approval. The ADM scaffold for wound care and hair regeneration, ADM paste for wound care and wrinkle and facial contour adjustment. And a cellular dermal patch, they're intended for hernia operation, lung operation, breast reconstruction, and durameter substitute. And they all have Taiwan FDA approval. And the porcine bone-derived product, we grind the porcine bone into granule, cut them into cube, stripe, wedged, and cylindrical bone. They are good for orthopedic and dental application in the bone void filling. And we also have this Lego bone material. You can put the pieces of bone together to form a lower jaw. And these are intended for the oral cancer patient, maxillofacial reconstruction, and also for cleft palate patients to do the facial reconstruction. And the bone graft putty for minor bone defect adjustment. And the cartilage graft for osteoarthritis, knee joint regenerations. And the bone graft material received US, Taiwan, Singapore, Vietnam, Philippines approval, and dental bone graft received Taiwan Philippine Titan, and the bone matrix for maxillofacial reconstruction received Taiwan and US FDA approval. And the easy bone or the injectable bone putty received Taiwan approval, and the cartilage graft for wound healing and for the knee joint cartilage repair also received Taiwan FDA approval. And the material have excellent biocompatibility. You can see that the osteoblast MG63 can attach on the bone material and proliferate and secrete new bones right on top of it. And so it was chosen by NASA to study the astronaut osteopenia. In a product under development, we have this cornea product. According to WHO, there are more than 20 million people suffer from the cornea blindness. When we found that the eyeball size of pig are similar to the eyeball size of human, and also the histological structure with the epithelium, keratocyte, and endothelium are identical among mammals. So we purchased the porcine eyeball and removed the cornea and decellularized by supercritical CO2 process, and it's ready for corneal transplantation. So we performed this canine penetrating keratoplasty in 2016. Pet dog Bonnie suffered from cataract and also corneal scar. So the right eye was diagnosed to be completely blind. And the surgery was simple to remove the whole cornea and replace the intraocular lens. And on top of that, put the porcine decellularized cornea, and 30 days later, the right eye becomes clear and visible. So this is the first canine corneal transplantation in the whole world. And Discovery Channel came to feature Bonnie's story. And this material has completed a human clinical study in Taiwan. And we recently received the export permit from Taiwan FDA. And we have these material, ADM scaffold, ADM paste for aesthetic medicine. The ADM scaffold are for hair growth, and the ADM paste are for wrinkle injections. And they all come from this porcine hide. And we slice it to become a membrane, and go through supercritical CO2 decellularization process. And after that, we cryo-grind it into a particle. The size range from 50 to 250 micron. And it's good for hair growth when you inject those particles under the scalp skin. And also, if you mix the particle with collagen slurry, put it in the prefilled syringe, it is ready for wrinkle injection. And the application of the ADM scaffold is to first mix with normal saline and resuspend it, and put it into the vital injector and inject under the skin. And you can see that after 30 days injection, you see a lot of hair growth. And also, this is another case, inject 30 days, you see a lot of hair growth. And this is one of our staff. He was injected every other month. And for the 150 days later, you see a lot of hair growth. And not only do you see new hair, you also see that the old hair becomes stronger and longer. And for the ADM paste, for the facial contour adjustment, like the tear trough adjustment, dark circle whitening in this case, and also for the eye bag adjustment, and for nasal labial fold and chin adjustment. Also, these are the tear trough and cheek adjustment. The physician are very satisfied with this material. And we also do cell therapy, which is combine the stem cell with our collagen matrix and then put them on the wound bed. The wound here in 18 days, and you can see that the reconstructed skin with epidermal, dermal, and also hair follicle structure. If you do not use the stem cell, just the collagen matrix alone, you do not see the same skin structure. So we can regenerate the whole skin within 18 days by combining mesenchymal stem cell with collagen matrix. In the cartilage graft, we grind the porcine cartilage into granule, go through decellularization process, and use that to do the knee joint cell therapy. First, we created a defect on the knee joint cartilage, and then put the decellularized cartilage graft together with PRP and chondrocyte, and mix together, put it in the defect area. 30 days later, we harvest the area and check on histology. You can see that the whole defect area has been reconstructed with the cartilage. And if you enlarge, you can see each lacuna has got chondrocyte inside. And if you look closer, this is the original cartilage, and this is the graft material we put in. And they are almost identical after 30 days. So we can reconstruct the knee joint defect by this method. And our ultimate goal is to do organ engineering by reconstructing heart, kidney, and liver in vivo. So what we do is to decellularize the heart, liver, and kidney by supercritical CO2, and then we put this decellularized organ scaffold back into live animal, connect the blood circulation, so the blood flow will bring in all the stem cell required for the organ reconstruction. And after it is fully reconstructed, it will be ready for organ transplantation on autologous basis. And we are successful in the rabbit kidney reconstruction model. So what we did was to decellularize kidney, a rabbit kidney, and then grab another rabbit, remove one of the two kidney, and then put these decellularized kidney scaffold on the same spot, connect the blood circulations. So you can see that blood flow coming to this kidney after surgery. And then you close the wound, waiting for the reconstruction to occur in vivo. And we reported this in the Formosa Regenerative Medicine Conference. And this is Dr. Fu Yinzi, who performed the kidney transplantation for us. So after putting this decellularized kidney into another live rabbit for 4 weeks, we sacrifice the rabbit and harvest this kidney and check on histology. You can see that it's already got a glomerular structure after 28 days. And we also stain with stem cell CD34 marker, and also the other marker showing that the stem cell has differentiated into many different cell types in the kidney. And also by checking with nuclear medicine, we see the reconstructed kidney already show urine inside the kidney. So that means this kidney is partially functional now. So we are excited to wrap up all the data and submit it a manuscript to Nature Medicines. And we also have these material for research use only. They are decellularized scaffold and put them into petri dish so scientists can culture stem cell or somatic cell for a regenerative medicine study. And we also grind those material into powder so that scientists can use those powder to do the 3D bioprinting. And here are some of our publication and submitted articles. And we have these trademark and brand name registered all over the world, and certificates, US FDA device master file, and also for the scar care series, We have Taiwan CE and US FDA and FDA 510K, and Singapore, Philippines, Vietnam, Thailand, India for the high-end medical device approval, and Taiwan FDA approval. And awards, we received numerous awards in the past few years. And Discovery Channel came to feature Bonnie's story. We take this group photo. This is Dr. Cai Zhonghan from Tainan Qimei Hospital. He is a human eye doctor, but he performed the corneal transplantation for Bonnie in a pet hospital. And this too is the host of Bonnie. And you can see the YouTube link for this video. And Fortune also featured our company story in 2021. It was about our heart reconstruction in vivo. The title was Two Hearts Are Better Than One. And we have contract signing with Australian company OBM, Marubeni in Japan, also Tribe Service General Hospital, and DKSH in Vietnam. And our slogan, we continue to make the impossible possible. So thank you for listening.
Okay, so thanks for Dr. Hsieh's insightful presentation. So if anybody have any questions, please feel free to leave the question in the chat room. So we are continue to the next next topic. So let's